This is the first of a set of videos that I'll be making. They might be a little bit different from some of the previous ones. I'm trying to get rid of the lag problem with the audio, which some people have experienced on some of the videos. And also, having reviewed some of the notebooks that have already been submitted, I'm satisfied that the information on the videos is sufficient for you to be able to see what it is you need to see. So I'm no longer going to tell you which specific slides I'm using here, but rather I'll let you explore the slides for yourselves. So here what we're going to look at on this short video is the epiphyseal plate, which is this structure here and some of the material extending down here. And as you know from your notes, the function of the epiphyseal plate is that cartilage growth at the epiphyseal plate drives the growth and length of long bone, and it does this by the addition of new cartilage at the epiphyseal plate on one side and the conversion of that cartilage to bone essentially on the other side. So let's go up a little bit in magnification now. And just make sure that we're on the plate itself. And here, developing here, is the epiphyseal plate extending from here to here. Now the direction we're interested in looking is this direction. On this side of the epiphyseal plate we have some bone here, which is the dark pink staining material, and please feel free to take a look at this and to compare it, if you like, with the hyaline cartilage, which we can see here. Now, as we look at this, it should be immediately apparent that there are at least three fairly clear zones which you can make out on the epiphyseal plate. There's a zone along here, there's a zone along here, and there's at least a zone along here. Now, different textbooks divide the epiphyseal plate into different numbers of zones, and for our purposes we're simply going to divide it into this zone at the upper part of the epiphyseal plate here, which we'll call the resting zone, and then a zone beneath that in which the cells are proliferating rather rapidly. We'll call this, maybe along here, the proliferative zone. And then a zone in which the cells get larger and secrete cartilage, so this is going to be in this region here, and then eventually a zone of hypertrophy and cell death, where the cells become very enlarged and eventually die off, and then finally down here a zone of angiogenesis and ossification. Angiogenesis means blood vessel development, and ossification means conversion to bone. So let's take a quick look now at higher magnification of some of the zones and the regions which we're speaking about here. We'll begin on the proximal part of the epiphyseal plate, close to where the bone is visible, and here we see the resting zone, and in the resting zone we see chondrocytes in their lacunae, so-called resting zone because the cells here apparently appear quiescent. Just in from the resting zone here, we're starting to see cells in the proliferative zone. In the proliferative zone, what happens is cells divide by mitosis, and they do so sufficiently rapidly that they begin to stack up on one another, with the cells contained in their lacunae separated from the next lacuna just by a thin little plate of cartilaginous material, and they form these large stacks of isogenous groups of chondrocytes. Then the cells begin to secrete additional cartilage matrix and start to push one another apart, is how it will appear. And it's this synthesis of additional cartilage matrix which causes the thickening by interstitial growth of the epiphyseal plate as a whole. As the epiphyseal plate becomes thicker, what happens is the chondrocytes toward the center part of the epiphyseal plate become isolated from the blood supply because cartilage is avascular, and as a consequence the cells undergo hypoxia or lack of oxygen, so they begin to swell up. Here we see a lacuna and a lacuna. This is the remnants of a cell, the remnants of a cell, and these cells swell up in their lacunae and begin to die. And this is at the same time that this is happening, we also get secretion of a special type of collagen called collagen type 10. So it's secreted by cells in the growth zone and by cells in the hypertrophic zone, and collagen type 10 enables the cartilage to calcify or accumulate calcium, and this precipitates the anoxia hypoxia even further by making it more difficult for oxygen to diffuse through. As the cells die, they release their contents, and this results in the erosion of the little plates of cartilage that separated the individual lacunae, such that you now have large gaps or spaces in this calcified cartilage, and into these large gaps or spaces we have cells migrate in from surrounding bone marrow, groups of cells like this. Some of these cells go on to form blood vessels. There's a little blood vessel here, and some of these cells are osteoprogenitor cells and osteoblasts, which sit down on the calcified cartilage, which you can see here. The mauve lavender color is calcified cartilage, and these cells sit down on the calcified cartilage, and on the surface of the calcified cartilage they begin to secrete bone, which is a reddish pink color. And where you see calcified cartilage with some bone deposited on the surface, as in for example here, we refer to this as a mixed spicule, because this spicule of material is made of both calcified cartilage, the blue material, and bone, the reddish material. Interestingly, the new bone that's synthesized is remodeled almost as soon as it's being laid down, and here you can see an interesting looking cell. It's a reddish colored cell. This one has 3 nuclei, and this is an osteoclast, one of the bone resorbing cells that form by the fusion of multiple monocytes, and these are very common in the angiogenic zone and in the mixed spicule zone at the epiphyseal plate. Here's another osteoclast here. As we move further and further away from the epiphyseal plate, the structure of mixed spicules becomes even more apparent, with here again the red material is newly synthesized and calcified osteoid or bone material, and the remaining blue or purple material is the remaining calcified cartilage. So these are mixed spicules. And again here we see an osteoclast here, which is engaged in the process of resorbing both the newly synthesized bone and in fact the cartilage of the mixed spicule. And as we move a little further, you can continue to see this process in action.